Click for IB Biology. Here we're going to be looking at the different models for how enzymes operate. We'll look at the classic lock and key hypothesis, the induced fit hypothesis, and the more contemporary conformational selection theory. A PDF of this presentation is in the description below, where there's also a quiz where you can test your understanding of the work that we're about to cover. So this is a model of the classic lock and key hypothesis, and part of this is that the enzyme has a particular shape. So we'll recall that enzymes have complex, globular, often quaternary structures, and that small parts of the enzyme have positions often made up of just a few amino acids into which the substrate fits like a jigsaw, which is what we actually call they are complementary in shape. And this leads to the concept of enzyme specificity, which is to say that for the reaction of a substrate into a product, there's an enzyme that particularly can cause this and pick up that substrate. So it's one enzyme, one substrate type argument. When the substrate and the enzyme have combined, in D we have what's called an activated complex, and in that interaction the enzyme begins to weaken the structure of the substrate, catalyzing the reaction required in order to form the product, which is shown here. Now, part of lock and key hypothesis is to notice that at the end of the reaction the enzyme molecule is unaffected and therefore theoretically can go back and pick up more substrate, and that has implications for the concentrations of enzymes that are required to complete a reaction. This particular mechanism doesn't account for some of the experimental observations, and so it was that Koshland in 1958 proposed what's called the induced fit hypothesis, and then more recently the conformational selection theory of the 1990s. So let's take a look at those. So for comparison, I've left the lock and key model here, where there's a complementary relationship between the substrate molecule shape and the active site of the enzyme. With the induced fit hypothesis, the idea is that the enzyme is able to change its shape and fold around the substrate, creating this kind of a complex here, but the relationship is still specific. Only this particular enzyme can wrap itself around and change its shape to create this activated complex, so it's still got the idea of specificity. In more recent times, and with other experimental evidence, this has been adjusted. In this case, what we have here is the same enzyme, but what it's doing is constantly changing its conformational shape between one form and another. An enzyme molecule, a substrate molecule, which you can see here is complementary to this particular enzyme, selects this form of the enzyme, creates the activated complex, and then creates the product. So the new part is that an alternative substrate, perhaps with this kind of shape, might be able to select the second conformational form and catalyze a different product. So we would then have the same enzyme is capable of creating different products because different conformational shapes are selected by different substrate molecules. So that is quite different to the two previous hypotheses and conforms better with a lot of the experimental evidence and data, molecular imaging, which has come about since the 1990s. Below are appearing links to the next video in this sequence, along with the subscription button, of course, and links back to the playlist so that you can take a look at the other videos that we've produced in this Click for IB Biology series.